hello to all my champions out there. So I hope, my dear children, you have already attempted the mock test paper 2, and this solution is going to be for botany. So from question number 91 to all the 45 questions are there, it's from botany. Do not be confused. And if you're someone who's wondering, what is ma'am telling? Where are these questions from? So my dear children, we are giving you free mock test for NEET, and these mock tests are being made by the best teachers out there. So this is the first year we are doing something like that for the whole YouTube crowd. So we thought that we should be giving video solutions as well. So make proper utilization of this free time that is there, okay? And where will you find it? The link is given in the description box. You can go get yourself registered and get access to these 5 tests, and please make sure you are practicing it religiously, okay? Try it out religiously, no cheatings. Now the first question, the number of species that are known and described range between 2 to 3 million, 1.7 to 1.8 billion, 3 to 4 million and 5 million. What is the answer? It's a straight question, so this is a straight answer as well. It is 1.7 to 1.8 million, okay? So next question, read the 2 statements. They could be crystallized and the crystal consists largely of proteins, as in whatever you're talking about, they can be crystallized and the crystals would be containing most amount of what? Proteins only. Now they have nucleoprotein, as in they have nucleic acid and they also have protein, my dear children. And the genetic material is infectious. Whatever genetic material is there, my dear children, it is infectious. It can cause trouble to the other organisms which is affected by it. Now these features belong to bacteria, viruses, prions, viroids. No, it is just viruses, because you know viruses, they have caspid, which is actually made up of what? Which is made up of proteins, my dear children. Inside that they can have the RNA, for example HIV, which has 2 single-stranded RNA, that is highly infectious RNA that they are having, right? So their genetic material is infectious as well. What about the other options? Why not the others? So basically viruses, yes, because they are made up of simple protein coat and genetic material. There is no cell parts, okay? And bacteria, they are not, because they are living cells with cytoplasm, membranes, organelles, so it is too complex to basically crystallize them, okay? Now viroids, no, because it is just naked RNA that they have. They do not have the protein thing that is there. And if talk about prions, it is just misfolded proteins that they have, and they definitely clump, but they do not crystallize, okay? So that is the reason. So you have your answer, that is virus. Now which of the following groups of organisms are included under chrysophytes? Okay, which of the following group of organisms are included under chrysophytes? So you're talking about kingdom Protista, my dear children. Inside kingdom Protista, you have chrysophytes, you have a lot of other things. So you are asked, what are those things that are being put down under chrysophytes? Now these things do not have flagella and things like that. So we can rule out dinoflagellates. Dinoflagellates have flagella. Even euglenoids, they have flagella. Slime moths, they are fungi-like things, okay? Even though they are under Protista, but they are fungi-like things that they have. So only thing that they are left with is diatoms and desmids. So they are the ones that are included under what? Chrysophytes or chrysophytes. So kingdom Protista over here. If you see chrysophytes, that is diatoms and desmids, they are Protista. Here also Protista, all of them are Protista only. Yeah, habitat, they are found in freshwater and marine. It's mostly marine, it's freshwater. Differences, that is more easy to remember. So usually non-motile chrysophytes, okay, and chrysophytes, and my dear dinoflagellates have 2 flagella. Euglenoids have 1 to 2 flagella, and over here they have amoeboid movement instead of having what? A flagella. So there's like amoeba, they have pseudopodia to move around. So these are the things that you should be definitely knowing. So your answer should be diatoms and desmids. Next question. Sexual reproduction in fungi occurs by all the following except. So all of them are spores. So you might be saying, ma'am, sexual reproduction by spores, can that happen? Can that happen? Yes, there are sexual spores as well. There are sexual spores which are haploid in nature, my dear children, which are results of there is a zygote being formed. The zygote undergoes meiosis and forms sexual spores which are haploid. Now, my dear children, this are all sexual spores, okay? This is asexual spore that you are having. This is what? An asexual spore that you are having. So sexual reproduction in fungi occurs by all of the following except zoospores, which are asexual spores. Going ahead, in chlorophyceae, the pyrenoids are located in. What do you mean by chlorophyceae? Talking about green algae. Do they have pyrenoids? Yes, they have pyrenoids. And what is the structure? Where is it located? It is inside the chloroplast, my dear children. And they not just store starch, okay? They not just store starch, but they also store certain proteins. Now, I'll show you a line given in your NCERT just like that. So, most of the members have 1 or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein besides what? Starch. So, this becomes your answer. Works? Works. Next question. In bryophytes, main plant body is diploid. Is it? Biflagellate anthrazoids are produced. First of all, in bryophytes, main body is not diploid, it is haploid. It is what? Haploid. Always remember that. And it is haploid gametophyte body that they have. Okay, so main plant body is not diploid, it is haploid. Do they have biflagellate anthrazoids? Yes, they have, and these, with the help of these flagella, they swim around and they move and they reach the archegonium, which is the female sex organ that is there. So female sex organs are absent, wrong, they have archegonia. Fruits are always formed, that is a feature of angiosperms, right? So this is the only answer that is correct. Biflagellates anthrazoids are produced over here. Remember, they have what? The main plant body is haploid gametophyte body. Haplontic gametophyte body. Next, fusion between morphologically alike gametes is referred to as in morphologically, if you see from outside, they are going to be similar. What do you call this? Fusion between two morphologically alike gametes. Isogamy, anisogamy, oogamy or syngamy. The answer is isogamy. You call this condition as isogamy. So isogamy refers to the fusion of morphologically identical gametes. Okay, both the male and the female gametes are similar in appearance and function, which typically occurs in some algae and fungi. Now anisogamy refers to the fusion of gametes that are different in size or form. Typically, one gamete is going to be smaller and the other is going to be larger. This is seen in animals and the plants. Remember that. Now under that only you get oogamy, where you told that here basically they do not have same structure, right? So here you are telling the situation where basically the female gamete is going to be larger, the male gamete is going to be smaller, and the male gamete is going to be motile, whereas female gamete, it is going to be non-motile. And there is syngamy. What do you mean by syngamy? Syngamy is just a fusion of gametes. That is what you call as syngamy. So isogamy, my dear children, it is where you have both the gametes which are morphologically similar. Next, root hairs arise from. My dear children, in the root you have different regions. First you have root cap, then there is meristematic region, then there is region of elongation, then there is region of maturation. Where do you get root hair from? It is from the region of maturation. And if you're worried about the picture, I am going to show you over here. So this is a root cap, this is a root, the region of cell division, that means the region of meristematic region, that is where the cells are going to actively divide. Then there is a region of cell elongation, and then there is a region of cell maturation. Okay, from this is where you are going to get the hair, root hair from. Cool. So this becomes your answer. Next, X is a scar on the seed coat through which the developing seeds were attached to the fruit. Above X is a small pore called Y. Identify X and Y. So my dear children, when it is an ovary and inside the ovary you have the ovule. So you have the placenta. Placenta is attached to the ovule with the help of funicle. With the help of a structure that is known as funicle, and this point of attachment is known as hilum. So hilum, wherever is there, it leaves a scar there. It leaves what? A scar there. So the scar which is there, it is hilum. And now you're talking about a pore. What is this pore? Micropyle region, my dear children, through which the pollen tube is going to enter. So that pore remains like that only, and it is there for moisture and everything to pass through. Okay, so your X, which is a scar, it will be hilum. So it will be B. Only B has that, yeah. So it will be B, and my dear children, about Y, it will be micropyle. Works? Okay. Next thing, the region of stem where leaves are born. If you talk about the stem, you have certain nodes, and in between that you have what? You have internode. So these nodes are, my dear children, are areas where you get what? You get the leaves and everything. So these are what? Nodes. So your answer become node. Going ahead, the lateral roots arise from. Where do you get lateral roots from? My dear children, lateral roots arise from the innermost part. Just okay. So if you talk about epidermis, epidermis is outermost layer. Yeah. Then you have cortex region, and then you have endodermis. After endodermis, you have pericycle. After pericycle, you have the vascular bundle and things like that. Yeah. And then in some plant there will be cambium also. So pericycle is a region from where you are going to get lateral roots. Always remember that. Okay, so this is how it looks. So this is the pericycle. Epidermis is there, then you have cortex, then you have endodermis, then inside that there is pericycle. From the pericycle you are going to see there is development of lateral roots which is there. So this is the order that is there. Epidermis, cortex, endodermis, pericycle, vascular cylinder, as in you are talking about xylem and phloem. Together you will call it as stele, and cambium in some plants. Okay, where secondary growth is also there. So this becomes your answer. Okay, Next, the cell wall and dash may be traversed by plasmodesmata which connects cytoplasm of the neighboring cell. So what happens, suppose this is a cell and this is a cell. So outermost layer that you have is cell wall for sure, and in between that what do you have? You have middle lamellae. You have what? Middle lamellae. So you have to make a pore like this. Don't you think this pore should be transversing the middle lamellae as well? It should be transversing the middle lamellae as well? Yes, it should be. So can I say the cell wall and dash may be transversed by plasmodesmata which connect the cytoplasm of neighboring cells? It can be middle lamellae. Yes, it's not cell membrane, it's not Golgi, it's not nucleus, it will be middle lamella which is there. Middle lamella is also known as a cementing layer which is there between the cells. So the cells remain compact, it remains together. Okay, and since you want things to be transferred, that is why you have plasmodesmatal connections. Now, the cell organelle involved in the formation of glycoproteins. So, my dear children, proteins are being synthesized with the help of ribosomes which are present on the rough endoplasmic reticulum. So, can I say rough endoplasmic reticulum is there for production of proteins? Right, I can say that. Yeah. Now, if we talk about smooth endoplasmic reticulum, it is there for the production of lipids. Now, both of them, basically if I want to modify them, where do I go? I will be going to the Golgi apparatus. In the Golgi apparatus, these proteins and these lipids would be attached with some carbohydrates, like making it glycolipids and glycoproteins. Okay, making it glycolipids and glycoproteins. So cell organelle involved in the formation of glycoproteins is what? Golgi apparatus. Works? Cool. Okay. Next, potato and brinjal are two different species but belong to the same genus. Which family do they belong to? It becomes easy to remember the genus then. Solanaceae family is where they belong. Now the genus where they belong is Solanum. Is what? Solanum. So that becomes your answer. Next, the parasitic fungi on mustard is. Now this parasitic fungi is going to cause a disease which is known as white rust. Which is known as white rust in mustard. So what is that? A particular parasite? Mucor, Rhizopus, Albugo, Aspergillus. Your answer is Albugo. Albugo is a parasitic fungi that is going to affect what? Mustard plant. It causes white rust. Next, study the given flowchart of cyclic photophosphorylation and select the correct answer. Before I go ahead, I just know one thing. In cyclic photophosphorylation, there is only one system, photosystem, that is PS1. Okay, it happens in the stroma lamellae and stroma lamellae lacks PS2, so there is only PS1 which is involved in cyclic photophosphorylation. And I also know one more thing, that PS1 is known as P700 because it takes the wavelength up to that. Okay, so with that I can try to find the answer. So what is A? A is something that is taking up the light, so it has to be photosystem, right? Which photosystem? It has to be one, so I have two options, so I can cut down these two options. Now, basically from here, when light falls, what will happen? Electrons are going to charge up, yeah? It will go to the high state. So there should be an electron acceptor there. So this should be an electron acceptor. Both of them are the same. Now over here back, it is coming back. Okay, it will be coming back. And what is the C over here? What is the C, my dear children? Can I say it's P700? Yeah, it's coming back to the same thing. That is why it is known as cyclic photophosphorylation. So which is why your answer becomes this. Do you think, my dear children, over here ATP is being produced? Yes. Do you think NADPH is being produced? No. Always remember, there is no production of NADPH in the case of cyclic photophosphorylation. Next, which of the following is both a growth promoter as well as a growth inhibitor? It promotes growth as well as inhibits growth. So auxin is strictly a growth promoter. Even gibberellinic acid or gibberellin is also a growth promoter. Abscisic acid is something that is a growth inhibitor. Okay, it is a growth inhibitor, does not allow growth to happen. Okay, now talking about ethylene, it is both going to act like a growth promoter as well as an inhibitor. Why? Because ethylene helps in the ripening of the fruits and things like that, which is basically making it grow, yeah. And in the case of other things, okay, they help in inhibition also. They also inhibit, certain times they lead to abscission of leaves, they make the leaves fall down and things like that. So that is abscission, that is growth inhibition. So that also takes place, which is why ethylene, I will call this as both growth promoter as well as inhibitor. Majorly categorized as an inhibitor, but yeah. Also remember, it's a gaseous hormone. Now, the leaf base expands into sheathy covering on the stem partially or wholly in bean, pea, grass, mango. So in bean and pea, the leaf base, my dear children, forms a structure like this, which is known as pulvinus. which is known as pulvinous. What about grass? In the case of grass, my dear children, the leaf sheath is going to make a covering up over here. Okay, in the case of grass, it is going to make a covering. I'll show you this. Over here, you see? So there's a covering, right? So this is seen in the case of grass, the sheath. Okay, not mango. Mango, you don't see that. Now, what is the function of filiform apparatus in angiosperm embryo sac? So if you talk about the embryo sac, you have two synergids, one egg cell, two polar nuclei, and three antipodal cells, right? So over here, you have the filiform apparatus. What do? They allow the entry of pollen tube. They guide the entry of pollen tube such that the male gametes which are there in the pollen tube can fuse with this and this guy. So it brings about opening of the pollen tube, wrong. It guides the pollen tube into the synergid, yes. Okay, this guy, yes. It prevents entry of more than one pollen tube into the synergid, wrong. None of this is again wrong. This is the only answer that you have got. Next. Other than DNA polymerase, which of the following enzymes are involved in DNA synthesis? So restriction enzymes are the one that cuts, molecular scissors, right? Beta galactosidase is again an enzyme that cuts, that breaks down lactose into glucose and galactose. Okay, permease is an enzyme that will be letting things inside. It will increase the permeability of the cell membrane. DNA ligase, I will call this as a glue. Why? Because suppose there is a niche over here in between. It is the DNA ligase is the one that will be covering this niche in the case of DNA. So obviously, can I say that it is the one that helps in DNA synthesis? Yes, to a certain limit, but yes. So DNA ligase is something that will be having the same property, not exactly, but yeah. Okay, it helps in DNA synthesis. Next, during transcription in eukaryotes, heterogeneous nuclear RNA is transcribed by hnRNA. It is transcribed by what, my dear children? So you have three types of RNA polymerase. RNA polymerase 1, okay, RNA pol 2, and RNA pol 3, which you do not write like this, you write like, just a second, pol 1, 2 and 3. So first priority is to make rRNA, second is hnRNA and also mRNA, third one which is there, it is for tRNA and snRNA and things like that. Okay, so over here, during transcription in eukaryotes, heterogeneous nuclear RNA, talking about hnRNA, it is transcribed by what, my dear children? It will be RNA polymerase 2. So first priority always given to R. Second priority given to hnRNA and mRNA. Third priority to tRNA. Next, who suggested that in order to code for all 20 amino acids, the code should be made up of 3 nucleotides? Who said that? Was it Mendel? Was it Hershey and Chase? Was it George Gamow? Was it Griffith? Your answer becomes George Gamow. He said that if you have 4 nucleotides, basically if they come in doublet format, it can only give rise to 16 codons, but you have 20 amino acids, right? So 16 codons means you cannot repeat codons, right, for the same thing. So that will be wrong. So they had to be in a combination like this, where you can get 64 codons. In the 64 codons, one codon can code for, so multiple codons can code for the same amino acid, not a problem. But it's not like one codon is coding for multiple amino acids. No, multiple codons can code for one amino acid, that is fine, extra is fine. So this was okay. So George Gamow was a person who said that you have to have triplet codons. Next, which of the following is the beginning of translation? What happens in the beginning of translation? You have the mRNA, and then what will come and bind? Basically, the smaller subunit. You know, ribosome is the one that will be helping in translation. So ribosome has 2 subunits, small subunit and large subunit. So the small subunit is going to come and bind on the mRNA, and this is how we say beginning of translation happens. So when the large subunit of protein factory of the cell encounters an mRNA, wrong. Protein factory isn't talking about ribosomes, wrong. Okay, anyways, large subunit is not the one that comes and attached first. When the small subunit of protein factor of the cell encounters an mRNA, this is the case. When the small subunit of protein factor of the cell encounters a tRNA, wrong. When the large subunit, no. It is just this. Okay, so first basically mRNA comes, it attaches to small RNA, small subunit, okay, and then basically here comes the tRNA, and after the tRNA comes the larger subunit. Next, the stop codons are UAA, UAG, UGA, all of this. UA, UAG, UGA. All of this, my dear children, all of this are stop codons. What is a start codon? You have one start codon, and that is AUG, which codes for methionine as well. Next, dash is the term used for the part of the plant taken out and grown in the test tube under sterile condition in special nutrient medium during the process of tissue culture is. So dash is something, okay, dash is the term that I use for a part of the plant. I am going to take dash something, okay, grow in a test tube, and that's how I do tissue culture. What is that part known as? It is known as X plant. Now it is not a part of the plant, it is X. So that's why it is known as X plant, not implant, not both A and B. It will be just B, that is X plant. This becomes your answer. Okay. Next, biopesticides are the chemicals which are used to destroy the pest, the living organisms on the products which are used for pest control, the organism which destroy the crops, none of this. So basically, my dear children, if you are using pesticides, you are talking about chemicals. Biopesticides means you are not using chemicals. Okay, instead you are using organisms which would be feeding on other organisms, killing them, the pest, okay, killing the pest. So not the chemicals, but the living organisms or their products which are used for pest control. Can you think about a few? Can you think about, they are also known as biocontrol agents. Okay, they are also known as biocontrol agents. It's there in microbes and human welfare. Okay, for example, BT, for example, ladybird, and all those things. Now, orchid flower petal and female bee show what? So, female bee color and the orchid flower petal shows what? Shows what? So, it is showing something that is known as co-evolution. They are going to evolve together, my dear children. Actually, it's not just female bee color. So, basically, orchid flower and the bee which is there. So orchid flower looks like the female bee and the bee is the male bee, so it tries to pseudocopulate, and in that way it does the pollination. So basically both have to evolve together. That is how it occurs. So it is actually co-evolution. It is what? Co-evolution. Orchid flower petal and the female bee color shows what? Co-evolution, not competitive release, no parasitism, not all of this as well. If the question is orchid flower and the male bee also, it would be correct. Next, when the population density reaches the carrying capacity, the logistic growth curve is said to be. For example, the population is growing, it reaches this level beyond which it will not go. What do you call this? Asymptote. You call this condition as what? Asymptote. Okay. So this becomes your answer. Next, select the option that correctly identifies A, B and C in the given table. Eagle. What is the trophic level of eagle, my dear children? Top carnivore is a top carnivore. So I have two options. I will cut this off. Yeah. And now, earthworm primary consumer in what food chain? Earthworm is a detritivore, so it will be there in the detritus food chain. So again, I have two options. Frog, my dear children, in the grazing food chain, what is the trophic level? So frog, basically there is grass. Okay, there is grass, and the grass is eaten by what? Grass is eaten by grasshopper. Grasshopper becomes primary consumer. Now grasshopper is eaten by what? Frog. Frog becomes secondary consumer, which is why your answer becomes this. Okay. Yes. Next, the curve shown by human population is. Human population, my dear children, do you think it grows like this? Do they show exponential curve? No. They are the ones that show logistic curve. They will be growing only up to certain limit, because the factors which are there, there are limiting factors. You don't get everything in abundance. Okay, there will be limiting factors, which is why the curve shown by the human population would be what? What shaped curve? It will be S-shaped curve. Cool? Works? Next question. And what do you call this curve? A logistic growth. This is logistic growth, my dear children. And the formula for this is dn by dt is equals to Rn k minus n divided by k. Now, the annual net primary productivity of the whole biosphere is approximately dash tons of organic matter. What is the annual net primary productivity? It's 170 billion tons. What tons? 170 billion tons. This is just to confuse you all. Okay, net primary productivity is just 170 billion tons. Straight question from biodiversity and conservation. Okay, my dear children, going ahead with the 32nd question. Which of the following statement is incorrect for the given picture? This one. What is this? This is Selaginella, and it belongs to what? Lycopsida. The first statement itself is correct. Itself is wrong. That is, it belongs to Spinopsida. Wrong. It belongs to Lycopsida. Now, it is Heterosporous. Correct. It is the first terrestrial plants to possess vascular tissue because it comes under Pteridophytes. Yeah. And the main plant body is Sporophyte. That is also correct. Only wrong statement given over here is this. A. Okay. Now, next question. Where is it? In Calotropis and guava, the phyllotaxy, how is the whorls arranged, my dear children? Basically like this. So it's opposite to each other. That is what you call. So this is opposite, not alternate. If it is alternate, it looks like this, yeah. If it's whorled, basically all of them would be around at one particular area. Okay, so basically this answer would be opposite. For Calotropis and guava, it is opposite phyllotaxy. Next, ovary is said to be half inferior in which of the following condition? So there is half inferior, as in it is not inside something, it's like this, and all the other parts, my dear children, would be almost the same height. Okay, that is what you call it, half inferior, and this is the condition of perigynous plants. Perigynous ovary is like that. Hypogynous means ovary will be at the top, superior ovary. Epigynous means ovary will be inferior, and perigynous means it will be almost equal. So your answer would be half inferior, then it is this. Okay, if it is fully inferior, then it is epigynous. Now, next, polyarch vascular bundles generally occur in, polyarch means more than 6 vascular bundles coming together, grouping together. Where do you observe it? Monocot stem, dicot root, monocot root. What is your answer for this question? Your answer would be monocot root. Okay, this would be your answer. 36th question. Select the wrong statement with respect to the structure of a plant cell. Cellulosic cell wall is present inside the cell membrane, but the first statement itself is wrong. If this is a cell membrane, outside it, no, there will be cell wall. Inside it, why there is a need of cell wall, right? So it will be outside the cell membrane, the cell wall will be there. Here it is written that it is present inside, which is why it is wrong. Now, centrioles are usually absent. Do you think so? That is correct. In the case of plant cells, centrioles are usually absent. Yeah, and the large central vacuole is present, correct. Golgi apparatus performs the function of packaging materials in the cell. Yeah, they do modification and also packaging in the form of vesicles and things like that. Okay, I hope you know that. From them only you get lysosomes also. Okay, now, yeah, light is rarely a limiting medium. Light is rarely a limiting factor in nature except in. So except in means you're asked about in which organisms light is the limiting factor. So light is rarely a limiting factor in nature except in what? Obviously the one that does not receive much amount of light, plants in shade or in dense forest, for them light is a limiting factor. Because of that, photosynthesis would be less. So this would be the correct answer. Not maize, not sugarcane, not sorghum. It will be plants in shade or in dense forest. So this is just to confuse you. Light is rarely a limiting factor in nature except in what? Plants in shade or in dense forest. Going ahead. The enzyme that is not found in C3 plants. The enzyme that is not found, do you have RUBP carboxylase? Yes, that is the one. Rubisco is the one that fixes the carbon dioxide. So that is there. NADP reductase, that is also there, which helps in reduction. Yeah, ATP synthase, that is also there. So what is not there? What is not there in C3 plants? It will be PEP carboxylase, phosphoenol pyruvate carboxylase. Phosphoenol pyruvate carboxylase is found in Hatch-Lack pathway or C4 pathway, where they have two types of cells, mesophyll cells and also bundle sheath cell. Twice CO2 is being fixed, first with the help of PEP carboxylase, second time it will be fixed with the help of what? It will be fixed with the help of Rubisco. Cool. So in them you have it. In the case of C3 plants, you just have mesophyll cells, and this mesophyll just has Rubisco, nothing else. Okay. Next question. Match column 1 with column 2 and select the correct option from the codes given below. TCA cycle, that is tricarboxylic acid cycle. Right. What happens in tricarboxylic acid cycle? Can I say it is also known as, it's basically Krebs cycle. TCA cycle is also known as Krebs cycle, and it was put down by whom? It was put down by Hans Krebs. So this is going with A, goes with 2. A goes with 2, my dear children. Okay, A goes with 2. I have lots of options like that. I can cut these options. Now F0, F1 particle. F0, F1 particle, my dear children, they are what? F0, F1 particle. If that's difficult, let's go ahead with the next question. End product of glycolysis. After glycolysis, glucose is going to convert into what? Pyruvate. So C goes with 4. C goes with 4. Both the options have it. Again, we have to go ahead. Pyruvate dehydrogenase. What does it do? What does it do? Pyruvate dehydrogenase, my dear children, it helps in the process of basically breaking down pyruvate to what? To acetyl-CoA, and this is where carbon dioxide is being released, which is why it is known as oxidative decarboxylation. Oxidative because NADH is being formed, okay, and also decarboxylation because CO2 is being released. So pyruvate dehydrogenase with this, okay, and F0, F1 particles goes with inner mitochondrial membrane. F0, F1, you know, if this is the inner mitochondrial membrane, the F0 particle is here, F1 particle is over here. Got it? So that is also there. So B goes with 1. B would be going with 1. So A goes with 2, okay, B goes with 1, okay, C goes with 4, okay, and D goes with 3. This becomes your answer. Cool? Now, next question. GM plants, that is genetically modified plants, are useful for reducing reliance on chemical pesticides, obviously because you are making them pest resistant, so you do not need pesticides there. Enhancing nutritional value, obviously yes. To create tailor-made plants to supply alternative resources to industries, yes, tailor-made plants, because that means according to our wish we are making up a plant. So that is tailor-made plants according to what we require. That is also correct. So all the statements are correct. So can I say all the above? Yeah, all of the above becomes your answer. Next, 41st question. The proteins encoded by genes cry1AC and cry2AB control what? The proteins encoded by genes cry1AC and cry2AB controls what? So always remember cry1AC and cry2AB controls. First letter is C, so cotton. Okay, second letter is B, so bollworms. So corn borrowers also C and V, ma'am, C and B then. So cotton bollworms, it is bit longer. If you talk about corn borers, my dear children, it is cry2AB. It is what? Cry1AB, not 2AB, cry1AB. Cry1AB. So over here only one thing is there for corn borers. Corn borers, C for corn and B for borers. So always remember cotton bollworms, it is big, right? So that is why you have 2. Here it's just a small thing, that is why you just have 1, and that is this cry1AB. That is cry2AB that is involved over here. So over here the proteins encoded by genes cry1AC and cry2AB controls what? Bollworms, cotton bollworms to be specific. Next, bottle juice are clarified by the use of what? Bottle juice are cleared up with the use of pectinases, because pectin will be there, yeah, and also proteases, not chitinases. Chitin you don't find in the plants as in like that, okay. You have pectin, you have all the other things, cellulose and things like that, but not chitin. So basically it will be pectinases as well as proteases. So I can say both A and B would be the correct answer. Okay, this would be the correct answer. Next, correction of genetic defect by delivering a normal gene into an individual or embryo to take over the function of and compensate for the non-functional gene. One gene is not functional, so basically you are compensating it by putting in another normal gene out there. What do you call this therapy as? You call this as hormone therapy, vaccination, none of the above. It is gene therapy where you are providing the gene itself. Okay, so A is your answer. Next, 44th, at present about dash recombinant therapeutics have been approved for human use the world over. Okay, how much? 30. Okay, almost 30. And now in India, B of these are presently being marketed. So out of 30, something is being marketed, that is what? 30, all the 30s? No, it will be 12. So there are 30 which are being approved for human use, and out of that 12, my dear children, are being presently being marketed as well. Works? Chalo. Correct. Now, functioning of statin is based on allosteric inhibition, non-competitive inhibition, competitive inhibition, end product inhibition. Functioning of statin is based on what? Statin is what? Blood cholesterol lowering agent. Yeah. So what does it do? Basically the enzyme which is responsible, it is going and trying to competitively bind with it. Okay, that is the substrate will be there, the statin will be there, it will try to compete with the substrate to bind to the active site of the enzyme. So this is known as competitive inhibition. What do you call this? Competitive inhibition. So this is how it works, and statin is being produced by Monascus purpureus, not purpureus. Okay, purpureus. So with that, we complete all the 45 questions, my dear children. So now the zoology part will also be coming up, but I'll request you keep on solving. Let's see, when it comes, it will be uploaded. You have to watch it and it will be helpful for you. And if you think you like the video, do not forget Do not forget to subscribe to the channel. Wishing you good luck. Bye bye. Take care, and do let us know in the comment section also how was the class. Yeah.